Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss on different types of RNA and their function. As we know that the central dogma of molecular biology deals with the flow of genetic information within a biological system. It is often stated as DNA makes RNA and RNA makes protein. Now this RNA stands for ribonucleic acid. Unlike DNA, RNA is generally single-stranded polynucleotide chain. The nucleotide present in RNA have OH on their carbon number 2 in the pentose sugar, and they have uracil instead of thymine, which is present in DNA. Now this RNA is different types, and it is firstly divided into two groups, that is coding RNA and the second one is non-coding RNA. The coding RNA is messenger RNA, that is the mRNA, whereas the non-coding RNA are various types. First is ribosomal RNA, which is designated as rRNA. Second is transfer RNA, that is tRNA. Third is small nuclear RNA, that is SNRNA. Fourth is small nucleolar RNAs, that is snoRNA. Fifth, PV interacting RNA, that is pRNA. Sixth is microRNA, that is miRNA. Number 7 is long non-coding RNAs, that is lncRNA. Now these first two, that is the ribosomal RNA and the transfer RNA, are known as housekeeping RNA. Housekeeping RNA. Okay, so these two are housekeeping RNA, whereas these are, that is small nuclear RNA, small nucleolar RNA, pRNA, microRNA, long non-coding RNA, these all are regulatory RNA. Regulatory RNA. Okay. Now, what do you mean by coding RNA and non-coding RNA? Let's discuss. The coding RNA undergo translation to synthesize protein, whereas the non-coding RNA do not undergo translation to synthesize proteins. In human genome, less than 2% are coding RNA, whereas almost 90% are non-coding RNA in the human genome. So this coding RNA is very less in human genome compared to the non-coding RNA. Now let's discuss about the function of these different types of RNA. First, we will discuss the function of mRNA, that is the messenger RNA, which is the coding RNA. The mRNA corresponds to the genetic sequence of a gene. This made from DNA template during the transcription process. As we know that the in central dogma status that the DNA makes RNA. So from DNA, RNA is produced, and this process is known as transcription, and from this RNA, protein is produced. From RNA to protein, synthesis is known as translation. The mRNA carries the coding sequence for protein synthesis and are called transcripts. So these mRNA are known as the transcript. The mRNA carries protein information from the DNA in a cell nucleus to the cell's cytoplasm. Here, the protein making machinery reads the mRNA sequence and translates each codon. Each codon comprises 3 nucleotide base into its corresponding amino acid in the growing protein chain. Okay. Next is the function of tRNA, that is the function of transfer RNA. The tRNA is an adapter molecule. It serves as a physical link. So this tRNA serves as a physical link between the genetic code of mRNA molecule and the growing chain of amino acids that make up the protein. So amino acids are bonded together to make the protein chain or the peptide chain. Okay. The tRNA helps decode a messenger RNA sequence into protein. Each 3 nucleotide codon in the mRNA is complementary to the 3 nucleotide anticodon in the tRNA. Okay. So the codon which is present in the mRNA it corresponds to the anticodon of the tRNA. Now each amino acid has its tRNA. Each time an amino acid is added to the chain, a specific tRNA paired with its complementary sequence on the mRNA molecule, ensuring that the appropriate amino acid is inserted in the protein being synthesized. Next, the function of rRNA, that is the function of ribosomal RNA. rRNA is the catalytic component of ribosome. It is a ribozyme. In the cytoplasm, rRNA and the protein components combine to form a nucleoprotein complex called the ribosome. The rRNA is the location in which mRNA and tRNA are able to come together to synthesize the protein. Next, the function of small nuclear RNAs, that is SNRNA. It is contain 150 nucleotides. These small nuclear RNAs are transcribed by RNA polymerase II. Small nuclear RNA are always associated with a group of specific protein to form the complexes, which is referred to as small nuclear ribonucleoprotein in the nucleus. The primary function of this SNRNP, that is the small nuclear ribonucleoprotein, is to process the precursor mRNA, that is a pre-mRNA. Function of small nuclear RNAs, that is the SNORNA. It contains 60 to 300 nucleotides. These are components of small nucleolar ribonucleoprotein. So these small nuclear RNAs are the component of small nucleolar ribonucleoprotein. These complexes are responsible for sequence-specific nucleotide modification. Next, the function of microRNA, that is the miRNA. It is the small non-coding RNA, about 22 nucleotides, and most widely studied class of the non-coding RNA. This non-coding RNA, miRNA, is recently used in various research purposes. This microRNA is transcribed by RNA polymerase II. This RNA species mediate post-transcriptional gene silencing through RNA interference, where an effector complex of miRNA and enzyme can target complementary mRNA by blocking mRNA from being translated or accelerating its degradation. In human genome, miRNA are estimated to regulate the translation of about more than 60% of the protein coding genes. Next is function of PV interacting RNAs, that is pRNA. It contains 20 to 30 nucleotides. This RNA binds with PV subfamily proteins that are involved in maintaining the genome stability in germline cells. It also plays a role in the gametogenesis. Last is non-coding RNAs, that is long non-coding RNA. It is a heterogeneous group of non-coding RNA transcript. It is larger than 20 nucleotides in size and make up the largest portion of the mammalian non-coding transcriptome. It is estimated that more than 8,000 long non-coding RNAs encoded in the human genome. These are essential in many physiological processes. To date, various mechanisms of gene regulation by some long non-coding RNAs have been reported, whereas most are still of unknown function. Now, in summary, if we discuss the function of different types of mRNA, the messenger RNA helps to transcribe into protein. Transfer RNA link the amino acid to the codon and help build the protein in ribosome. Next is the ribosomal RNA or rRNA. It forms ribosome, which is essential for protein synthesis. Next is small nuclear RNAs, that is SNRNA, which helps in processing of RNA, that is in splicing and also in gene expression. Small nucleolar RNA, that is snoRNA. It guides site-specific rRNA modification. Next is PV interacting RNA, that is pRNA. pRNA helps in regulation of cellular genes. Next is microRNA, that is miRNA, which is very important. It controls gene expression mainly by binding with the messenger mRNA, that is the mRNA in the cell cytoplasm. Last one is the long non-coding RNAs. It regulates the gene expression and other cellular process. These are all about the function of different types of RNA. If you like this video, please like, share and subscribe to this channel. Thank you.